Hi, I'm Rick Young. I'm a professor at Whitehead Institute and MIT, and I want to tell you today why I'm especially excited about a new discipline in biology called condensates. I got into this because I work on gene control, and in gene control we have a number of enigmas that have not been solved by the assumptions I've had about how biology works. We've long had assumptions about cell biology, for example, that there are membrane-bound compartments that segregate particular proteins and RNAs and other components into particular parts of the cell to do specific jobs. Well, I've also long had the assumption that most of the functions of proteins are in the form of these structures that are rather static, change conformations to do their job. But some of these assumptions have come up against evidence that we need to think a little differently about at least portions of how the cell works. There are, for example, many organelles in the cell that do not have membranes, that incorporate and concentrate specific components of biological functions that apparently don't need membranes for compartmentalization. There is in addition evidence that many proteins have portions that are intrinsically disordered, that don't form stable structures that are the kinds of structures I have used historically to imagine how biology works. Instead now, as we look at how gene control works, and we look at the key components, the DNA binding transcription factors and the DNA elements they bind, we see that genes that play the most prominent roles in cell identity, genes that are playing the most prominent roles in disease, that instead of these models that I've long lived with, a new model's emerged that I think is well supported by recent data. This new model is that the protein and other components that are important for gene control become compartmentalized into condensates, into organelles that are not membrane bound. Inside these condensates, you concentrate all the key components you need to carry out a complex reaction, and this additional concentration provides key features to those genes that are most important for cell identity. These same types of condensates, but in other forms, in dysregulated forms, are occurring at genes that are most important for disease. And I think this new view, which has come from a rather small discipline of condensates, of phase separation, at least with respect to biology, has now ballooned into an interest of many investigators in biology. For me, there's a very important implication. The enigmas that have existed in my field of gene control are falling to this new model. They are being explained nicely by this new model of condensates and phase separation. And importantly, 40% of the proteins in our genome have these intrinsically disordered regions that indicate that they are not stable structures, that they may themselves be involved in other kinds of functional condensates. And so much of cellular biology may in fact be in this new space. But most importantly, this tells us that there is a new lens with which to view disease. Much of disease-associated variation is in the DNA regulatory elements for genes. Much of what is dysregulated in cancer occurs in this space. If much of disease is involved in these types of condensates, then it also, importantly, tells us there may be new ways to develop therapies for patients with the most difficult to treat diseases.